Maybe by February and we'll get the notification. I think in April we may have exam. Like we may have exam. I think April or May. Expected actually. Really expected. Okay, that's why I'll finish syllabus by March 15. If any doubts, you can regularly will be in touch. Okay. Uh, After March 15th, like I'll finish the because I have completed 2 units already. Remaining 3 units, it's very simple units except quantitative genetics. And and actually, if they have active live participant, na, the class will like very like it will be more discussion. Like nowadays, there it's more one-way teaching. That's why I'm going very fast nowadays. Then, like when a Mendel's, like when we come to topic, the forward and reverse genetics is nothing but many people will think like it's something other thing than the genetics and plant breeding. It's not like that. It is just the terminologies. It uses the same technique that we have already discussed, and today we will give that idea about how what is forward genetics and what is reverse genetics. See, when Mendel started the genetics, it is forward. Means that's why it is the direction in which Mendel started the study of the trait, it is called as forward genetics. For example, if Mendel started in the other way, then that would have called forward genetics. It's not like that. If you go in that only, that is forward. If you go in this only, it is reverse. It's not like that. Which was the first? Like Mendel actually studied the traits. Then first he observed the garden, like in a Mendel, like in a garden, tall plants, dwarf plants. First he observed the traits. Then he thought that what is the nature, why these traits are so much different, and how it passed to the next generation. Like that's why he laid out the subject genetics. Then that is called as forward genetics. Means first you study the trait, then you are going for what is the cause of that phenotype. What is the cause of the trait? That is only forward genetics. What are all the Mendel's trait? That is forward genetic. And what are the we readers normally do? That is forward genetics. Okay, forward genetics is not nothing a new technology. Okay, just already present from phenotype. You're going to the genotype. That is only is forward genetics. But why this another term reverse genetics came to existence? For example, in when 1980s, when the DNA based technologies have been in a front line, like recombinant DNA technology or DNA structure as 1943 DNA structure came, and in 1990s DNA sequencing is taking has been like has taken very glory. Then in 2000, Human Genome Project was started. Means the sequence of the gene like available for every crop. Like nowadays, almost all the crop we have the sequence. Then the question is, we have complete sequence of the particular organism. But see, sequence means what is nothing but just an order of nucleotide. But either the which part of the that sequence will govern for which trait we don't know. Either how many genes are present in that sequence we don't know. Either which sequence is responsible for which trait we don't know. First, we are already having a sequence. We want to know that sequence is responsible for what in the phenotype in the organism. That is called the study is called as reverse genetics. Means we have a sequence information, but we want to know what that sequence will cause the effect in the organism. For example, you have a rice genome. We have a rice genome. We have continuous sequence like for the TMB sequence we have like this ATGC, ATGC. But which part of this one? For example, this to this one. We want to know, okay, here, this is a gene, we can identify the gene also. We can predict, okay, this part of the sequence is a gene. How we can predict this part of the sequence as a gene? Can anyone? For example, we have a continuous stretch of nucleotide. We can say that now, as per the bioinformatics advanced, like science advanced, can able to say, okay, from this part to this part, okay, this is a gene. This is a coding gene, coding part. How we can say? And we don't, I'm not telling it is only that it will go on for a rate. I'm not telling. Just we can say, okay, this is the coding sequence. How we can say? It is called as gene predictions. We can
Start
predict.
codon to stop codon, sir.
We have gene means some structure it should have. Either it having start codon, end codon. Like start codon means AUG, and termination codon is, so the three codons.
UAG, UAA, UGA.
Yeah. Okay. Then, and we have another structure like promoter also we have it, and also we having continuous this one. And if it repetitive genome means, and most of the coding sequence are not repetitive. Based on several points, we can predict, okay, this sequence may be exon, this sequence may be intron, this sequence may be repetitive, satellite. Based on that, we have prediction. That is one technique called gene predictions. There are a lot of gene prediction technologies over there, homology based gene prediction technology and de novo based gene prediction technology. Okay. We have all the information. We have very good bioinformatics tool to predict the gene. We have sequence, everything we have. But we want to know that particular gene is governing for which trait. That we don't. We want to know what else we can do. We already discussed many techniques.
Or we can silence that or upregulate or downregulate.
Yeah, yeah, we can silence that particular gene. We have a sequence. In which way we can able to silence this gene?
RNA interference.
We have discussed RNA interference. That is also one reverse genetic technology. Okay. Then another one.
CRISPR, sir. CRISPR.
Yeah, CRISPR. That also we discussed. That is also reverse genetic. Any other ways?
Methylation.
What?
Sir, I just thought methylation can
Yeah,
also.
methylation. That also we can do. Adding the methylation markers that we can do. But that is very difficult. Transcription level gene regulation. That also. There is another technique, virus induced gene cells. It is just like siRNA techniques. Virus induced gene cells. Then you can also see not only silencing. If you this is called all these technologies you told is it is loss function mutation. Means where by loss function means if you silence the gene, there is a loss of function of that gene, so that some trait will going to miss. Based on that, we can study. For example, there is a one gene called as FT gene, flowering locus gene. FT gene. It is actually produced in the shoot and it will go for leaf and it will go up to apical meristem and it will start flowering. That FT gene. For example, you know this gene sequence, but you don't know the function. What I can do, I can mutate this FT gene either through RNA interference or through CRISPR or through virus-induced gene silencing. We can mutate. Then, if you mutate the gene, what is the nature? What the plant? What is the nature of plant will become? I told what is the function of FT gene?
Sterile, sir.
Yeah, it will not produce any flower. Means, for example, you take one gene sequence, you just mutate it, the particular plant is not flowered. Means you can say that, okay, this FT gene is responsible for your flowering. Means by losing the function of this gene, we can able to predict the we can able to predict the phenotype which governs by this particular gene. This is called as loss function mutations, either by RNA, CRISPR, virus-induced gene silencing. There are another technologies we will discuss in coming slides. Okay. Then another one is gain function mutation. It is asked in the exam. Okay, don't neglect what is loss function mutation, what is gain function mutation. Most of the mutations are gain function mutations or loss function mutations.
Loss function.
Loss function mutation. Okay? And in rare cases, we will get gain function mutation. What is gain function mutation? Just reverse of this. If the particular gene is there, the function is there. For example, oxygen synthesis. For example, gene A is responsible for oxygen synthesis. You want to confirm this. How you can do? For example, you just increase the number of genes, gene copies, gene duplication, or whatever you can make, or overexpress the gene. There are several tools are there to overexpress the gene. Can you, anybody knows? You can recall? Yeah, correct. Riboswitch. How we can overexpress the gene? There are several technologies that we will discuss. Either one through gene duplication, increase the copy number of this particular gene A. Then, for example, if there is only one copy of this gene, for example, it produces some picogram, 1 ppm of your oxygen. But if you increase nearly 5 to 6 copy, then definitely it may increases 5 ppm of oxygen. Means increase in the concentration is directly proportional to increase in the copy number. So that we can predict, okay, this particular gene is involved in the oxygen biosynthesis. This is called as gain function mutation. Means if you are doing something that increases your phenotypic expression, that is called as gain function mutations. And gain function mutations, there are one technology famously used used in larger scale, that is called as activation tagging or overexpression by increasing activation tagging, or we can modify the promoter modification. Like, can anybody heard about the two terms, strong promoter and weak promoter term? What is strong promoter? What is weak promoter?
Yes, sir. Hard.
Yeah. What is strong promoter? What is weak promoter?
Strong promoter will activate and more higher influence the genes, sir. That means it will activate it in more expression. More expression will be there by the strong promoter.
For example, the same gene, gene A and gene A. If it is with a strong promoter, it will produce so many number of copies of mRNA. It is a weak promoter. If it is attached with weak promoter, maybe it will produce only 1 or 2 copies of the RNA. Okay. This is called as this when there is a higher expression, like when the higher production of mRNAs, when it is associated with a strong promoter. When it is weak promoter, it will produce very less quantity of RNA, with lesser expression of the particular gene. If you change, for example, if you replace weak promoter with a strong promoter by any your transgenic technology or insertional mutation, whatever strong gene transgenic technology, we are just modifying the promoter. We are increasing the function of the gene. Then we can study the phenotype. That is one technology. That is called as either activation tagging, means you are modifying the promoter in such a way so that the gene will express in a higher quantity. That is one activation tagging or promoter modification or increasing the gene copy number. There are any number of ways we can increase the function of the particular trait. That is called as gain function mutations. Either we can study the gene either through loss function mutations or gain function mutations. And you should know what are the technologies or tools are available to create the loss function mutations, and you should also know what are the tools and techniques available to study the gain function mutations. Understood? Can anyone summarize what are the tools are there to create loss function mutation?
Sir, for loss function, that is RNA interference, and this CRISPR
Yeah.
and gain function. We can use the genetic engineering, and we can replace the weak promoter with the strong promoter.
Yeah, that is called as activation tagging or increase. Another one, how we can increase the expression of the gene? How the, if the gene expression, for example, in transcription, what are the tools, what are the components we need for the transcription?
Promoter,
Gene want
sir.
to express, okay, promoter,
Transcription factors.
yeah, transcription factors,
RNA polymerase.
RNA polymerase,
Regulatory elements.
what are the regulatory units? Enhancers.
Yes.
This is one of the
Regulators.
enhancers, activators, mediators, regulator, okay. But majorly enhancers should be there, promoter should be there, transcription factors and RNA polymerase. For example, either you want to modify the expression of any gene, either you can modify it any number of components. Either you can modify the promoter, you can use strong promoter instead of weak promoter. Either you can add some transcription factor so that, for example, increase the transcription factor so that gene expression will be high. And RNA polymerase, no, it is if in the same quantity, higher quantity, it will go on by and it will start the polymerase like this one. And you can enhancers. You can increase the enhancer. Like for example, if the gene don't have enhancer, that you can introduce the enhancer. Okay, so that the gene expression will be very high, either through modification or introduction of enhancers, or modification through promoters, or modification of transcription factors, or like in RNA polymerase, so you can use high precision RNA polymerase. By all this means, we can improve the expression of the particular gene. Okay, there are several technologies. I'm not going in detail quantitatively. There are promoter trap, enhancer trap, or gene traps. There are several things are there to study the function of the gene. Okay, either you can use enhancer traps, you can use promoter trap, either you can use gene traps for study the particular function of the gene. Okay, then what is the difference between forward and reverse genetics? We'll come. Okay, we'll have some techno. What are the tools? Forward genetics is approach of determining the genetic basis responsible for a phenotype. We having a phenotype, we want to know either which gene is responsible for that. But a reverse genetics is approach used to understand the function of the gene by analyzing the phenotypic effects of specific engineered gene sequence. Means what I have discussed, mutation, either creating loss function or gain function mutation. Means here the procedure will start from phenotype. We first screen the population, we observe the phenotype, we check the how much variation is there, then we start like how we do breeding, it is just similar technique. But here we have first we have sequence, then we start modifying this sequence, then analyzing the phenotype. Then it is involvement of identification of the gene responsible for the particular phenotype. But here, identification of function for particular gene by creating mutations. Then, what are the forward genetic tools are available? We have discussed in detail GWAS, it's a forward genetic, QTAL mapping, it's a forward genetic, and your map-based cloning, it is also forward genetic. QTAL-seq is also forward genetic. Please watch my video about QTAL-seq if you are interested. MITMAP and MUTMAP plus also forward genetic. These are all NGS based method to identify the mutation. For example, you are creating a mutation in a plant. You want to know where that mutation is created. Means mapping the mutations. These techniques are used to map where the mutation is occurred in the genome through NGS technology. Just remember this. How we can do, what is MUTMAP, what is MUTMAP plus, what is MUTMAP gap, what is SURE map. This is not required for you. Okay? This you will study in genomics in plant breeding in PhD. Then there are reverse genetic tools. There are several tools that we have already discussed, like RNA interference and CRISPR-Cas9 and insertional mutagenesis. We will discuss this one. What is insertional mutagenesis?
Sir, we can able to track the place of mutation as well.
No. Just I'm telling insertional mutagenesis. What is insertion?
Addition of few bases.
Yeah, addition of genes or few bases, whatever it may be, through your transgene. For example, you are inserting transgene into this genetic background. Means if you are introducing transgene into this DNA, somewhere it will come and bind. For example, here it will bind, you think. Means in the original sense, in a before this introduction of this transgene into this region, then definitely some genes will be there here. Okay, that function will be altered if this transgene is inserted in this one. Definitely the genes present will be here. It will be silenced or removed. Then the function related to those genes will be altered. It is one type of loss function mutation. Means you are creating loss of function mutation by introducing transgene in the genome. Means you are inserting a foreign transgene in the genome and we are altering or we are removing the native genes and we are creating loss function of mutation so that we can study the function of that particular gene. That is called as insertional mutagenesis. We studied in detail about lag gene or antibiotic resistant genes, insertional inactivation. How many of you remember? Selectable marker, insertional inactivation. What we will do in insertional inactivation? In transgenic technology or recombinant DNA technology? Yeah, is correct. Either you can take XGAL lag Z function. Here, for example, lag Z is there. In this place, we are introducing some transgene, means the function of lag Z gene will be disrupted. So that we are not producing any beta galactose enzyme, so that it will turns into white colonies only. That's what we have discussed in detail. In the same way, we are inserting a transgene and we are creating loss function mutation so that we are studying those genes are responsible for which trait. That is only your insertional mutagenesis. But one thing you can ask me, I told this insertion is random is procedure, random in process. We don't have a control where this transgene will go and bind in the genome. Then how can you know that in this particular gene only the mutation is happened? For example, in this particular region only mutation is happened, and in this particular gene only mutation is happened. How we can know? How we can track that?
We will compare before sequence and after sequence.
Yeah, that we can do. That is, for example, you are introducing nearly 1 kb or 2 kb, maximum 3 kb gene. See, you are having 450 million base. For example, you take 450 MB genome. If it only 10 kb inserted, how you will come to know? All bases are, see, even after introduction of gene, it is also a nucleotide sequence. How do you compare? It is too difficult. You can compare, okay, I'm telling you, you can compare, but it's too difficult. Any other options?
We can locate that gene by tagging it with the fluorescence, sir.
Yeah, that one thing you can do. There we can track where at what location your like in which tissue this will happen. We can locate. Okay, for example, it is the gene activating in a flower. Okay, this fluorescence is there. Okay, this gene is responsible for flower. We can locate the where the particular gene is expressing. We can locate. Okay, if you can have a fluorescent tag. But where in the genome it is located, how we can? Have you heard about inverse PCR or tails PCR or a plasmid rescue technologies? This is not required for you, but I just for a curiosity purpose I'm telling you. Okay, there are several technologies are there. For example, anyhow we know the sequence of this transgene. There is a one technology called as inverse PCR. Means if you have only one primer also, like first it is in linear, then it is circularize the DNA by using one primer. Then after that there is a one technology called as plasmid rescue. Means if there is any circular DNA was there, it can be isolated. Like you just remember inverse PCR and plasmid rescue. From that we can able to track where your particular mutation is happened, where this particular transgene is inserted in the genome, that we can track. Inverse PCR, or another one is there, tail specific PCR, TAIL, thermal amplification iso, TAIL PCR, and inverse PCR. You just remember these two PCRs, tail PCR, inverse PCR, to track where your gene is inserted into the particular sequence. Okay, then that is about insertional tag genesis. Then virus-induced gene silencing, it is just like siRNA technique. You are introducing a double-stranded gene RNA into the plant. Means many of the viral genome is RNA based. If you're introducing, means it will go and bind some degrade some mRNA, so that if mutation will occur, we can study that gene. Then another one is tilling. Tilling is very important, that I will going to discuss. In breeding also it is very important. It is very helpful in the allele mining. Tilling is very important technology. Actually, many of years we are still using this, and it's very important reverse genetic tool. For example, we know the gene. We know the gene. we want to know how many number of alleles are there for this particular gene. Normally in plant breeding, how we can use stilling means I'm telling. For example, land races are there. For example, 100 land races were there. One important gene is identified for high quality, like aroma. You just screen all your land races with this gene and you identified alleles present in this gene. Nowadays, nobody is using because NGS is there. We just go either GBS or whole genome resequencing. We can identify the mutation. But earlier, for example, you take wheat, whole genome resequencing is very, very costly. In that case, we can use for cost effective method, we can use stilling to identify alleles of, we can mine. We are identifying how many number of alleles are present in this mutant land races, sorry, in this land races for particular this gene. If you want to know that, you can use stilling. But no, you can also identify the alleles in so many ways. Like you can go for whole genome resequencing, or you can go for GBS sequencing, or anything you can do. Normally, we will prefer nowadays whole genome resequencing to identify the allele. Last time we are told like whole genome resequencing we can also do to track the transgene, but it's very costly. If the genome size is very small, you can go. But if the genome size is very high, then identifying the alleles for a particular gene is a tedious task. Like that tilling was majorly used technology means that what is it tilling means? Like as I told, first you just take the DNAs from all your population like land races or mutant population. We just do pooling. After pooling, we know the gene sequence. I told for tilling we should know the sequence of the gene. Then design primer for this gene specific primer. Then you amplify this pool of DNA with gene specific primer. Then your PCR product will be generated. Then I told like when we are fooling, then definitely there are from different DNAs, so there will be some mutant will be there. You just denaturate this PCR product, then denaturation, then again you renature. While renaturation, what will happen? If there is any mismatch, see T should be there, but here G will be there. Here also some mismatch means there will be formation of heteroduplex. There will be formation of heteroduplex. There is a one particular enzyme called as cell1 enzyme. What is the function of this cell1 enzyme is to break this heteroduplex. Means if this heteroduplex is broken, then definitely there will be two fragments will be coming. Instead of long fragment, you will get two fragments. Means definitely here, see if it is break, means here one fragment will come, here one fragment will come. This smaller fragments we can able to detect. See in this sample, we have two fragments, but in this sample, sorry, three fragments. In this sample, only one single fragment. By this, we can able to identify the which nucleotide in this one, which nucleotide is responsible for mutation and how this nucleotide is varied among across the population we can able to study. Because each DNA we are pulling and we are doing this analysis when we can able to identify okay which allele is present in this all the lesion, and here you can able to study okay which allele is responsible, which nucleotide is responsible for this mutation, that we can able to study. This is means targeted induce targeted inducing local lesions in the genome. Means we have a target, then we are inducing lesions, means these lesions in the genome. Means we are mapping this particular region or a nucleotide in the within the genome by using cell1 enzyme. This is called as targeted induced local lesions in the genome. Understood, Tilly?
Yes,
Yes.
sir.
Then
Sir, after
board,
gel
yeah.
electrophoresis, we will sequence that, sir,
Yeah,
or
that
we will
nowadays,
by ladder
nowadays
or
we can
something.
do. Nowadays we can do. Okay, nowadays we can sequence. But there is a actually in normal gel we can't run. There is a
Sir,
one.
you
There is
told that
a
exact base pair we can detect at what location
yeah.
there is
See,
mutation.
nowadays you can
Without
just take this one, we can sequence so that we will get okay which nucleotide is there, that we can do. But earlier days sequencing was not there. Na? Earlier they just okay here there will be mutation. Can go for Sanger sequencing and we can identify which nucleotide is there. That also we can do. Yeah. But here we are going to get okay where is in this whole entire gene where is that particular nucleotide is causing the mutations so that we can able to study. There is another called echo tilling. Instead of here we are using mutations, but you can use naturally present mutations and pull it, and rest of the procedure is same. Then this is about the different technologies, and in SRF, see what are the things I'm discussing from last few days. There are only one or two questions that you may expect or may not expect. Don't go in detail about these technologies. Just because we discussed all these QTL mapping and GWAS and the association mapping. Okay, you just know. Okay, this is forward genetics because here we're starting with the phenotype, we are ending with the genotype. You should know we are studying about siRNA, we are studying about miRNA, we study the CRISPR and all those things. You just know that okay, these can be used for studying the gene. Like this approach is called as reverse genetics. Rest of it, don't worry. Okay? Nobody will going to ask in the exam, but you should know what is stilling, what is gene silencing, what is CRISPR, what is virus-induced gene silencing, what is insertional mutagenesis. At least you should know the definitions. Okay, but you should know in detail about forward genetics because it is integral part of plant breeding. The QTL analysis, GWAS, association mapping, and QTL-seq, RNA-seq that we have discussed in detail. In QTL-seq, watch my video. You're going to get. Okay, if you know this much, it's more than enough for the SRF. Then discussed about RNA-seq. There are other technologies than the RNA-seq and for gene expression analysis, northern blotting, real-time PCR, and differential display RT-PCR, and SSS technology. Actually, this is where this one is, and cDNA AFLP technology, RDA technology, SACE technology, microarray, and RNA-seq. But don't worry, nobody is using all those technologies. Only RT-PCR and microarray RNA-seq is majorly used, and RNA-seq we discussed, and RT-PCR I will discuss today. What is RT-PCR? In the same way, we have to isolate RNA from treated and untreated. Then you go for reverse transcription. Then you create cDNA. Then you do PCR. But PCR we have to do in a different way. Here we have to use a primer and along with the dye. There is a dye called as there are two types of dye. There is a like a scorpion dye also we can use, or you can use another type of dye. That name I forgotten. Cyber green dye. So this is called a cyber green dye. Two types of dye you can use: scorpion dye or cyber green dye. So that whenever your PCR product is formed, this fluorescence will going to observe. Based on this fluorescence detection, we can in real time basis we can quantify the gene expression. That is called as RT-PCR. Okay. Nowadays in every lab we are using RT-PCR. Then this function of gene can be studied through subtracted cDNA libraries, that is your SSS only, subtracted cDNA libraries only, whole genome sequencing. Through whole genome sequencing, we can't study the function. We can only know the structure of the genes. A, C, D will be the answer. Okay. Then we have discussed RNA-seq in detail. So I'm not going in detail. Any doubts? I hope I covered all the Unit 5 syllabus. I have covered all the Unit 2 syllabus. If you please go through again the syllabus, if anything needs, please text me. Don't hesitate. Okay. I will ready to
Yes.
take that one. Then tomorrow, from tomorrow, maybe I will take very lengthy classes because it needs a lot of time. I will go to. I will discuss PGR first. So I will at last I will discuss quantitative genetics. Okay, I need to be rearrange all the quantitative genetics slides. Actually, this is not enough. As for the last year paper, this much is not enough actually. To in detail. Okay. Tomorrow we
Some
will
questions
tomorrow
are
I will
there.
discuss. Yeah.
Some questions are there, I think, sir. Can you
No,
discuss?
earlier I used to teach for every topic. I used to take questions. Okay.
No, sir. In the slides, some questions are there, na, sir. Can you ask?
Yeah. In the slides, like after discussing topic, we have to discuss. Like every topic, I used to discuss after discussing the topic. Like heritability, the questions related to heritability, and breeding value, the questions related to breeding value. GCA and SCA, question related to GCA, and diversity analysis, questions related to them. Mating designs, questions related to them, and what else is? See, and I
Are
should
there
compare in
any
a very
questions of reverse genetics, sir?
nothing. I think nothing. No, yesterday we have discussed. No? This one we have discussed yesterday. No questions. See, nobody will going to ask actually. See, reverse genetics and this is CRISPR based. If any questions came from CRISPR based, is just definitely it will be a questions on reverse genetics only. There is no specifically. It is just there in the syllabus. You should know what are the tools will go under reverse genetic, what are the tools will go under forward genetics. It's not mean that these tools cannot be used for forward genetics. It's not like that. Just what is reverse genetics, what is the definition you should know, what tools are available for the reverse genetics. And it's not mean that that tools cannot be used in forward genetics. You should not think like that. It's not exclusive in nature. They are inclusive in nature. Okay. Okay, bye bye. Tomorrow we'll meet. Tomorrow we'll start the PGR. Okay? PGR I will cover all the sections of the PGR. I will remaining part cytogenetics will. Bye bye. Good night.
Good night, sir.
Good night, sir.